Humans, like most other organisms, have an internal biological clock that controls sleep and other daily cycles. It's this internal clock that is responsible for the feeling of jet lag often experienced on long flights. Hormones such as melatonin may be involved in this bodily reaction, as melatonin levels rise during the night and fall during the day. Such daily cycles are called circadian rhythms. Circadian rhythms are responsible for nocturnal animals searching for food at night and diurnal animals foraging during the day. Circadian rhythms are genetically determined. Mutations in the fruit fly Drosophila have been identified that alter the fly's circadian rhythms. Recent work has also identified the first such circadian rhythm gene in humans. Plants, like animals, have circadian rhythms. They open and close their flowers, their leaves, and their stomata in response to external cues such as sunlight and darkness, and internal factors such as the flux of calcium ions. Plants also have a seasonal cycle. External cues like temperature, water, and sunlight affect the levels of plant hormones that control the plant's growth and development. Changes in the environment may put a plant under stress. In response to stress, hormones signal for changes in the plant's metabolism and structure, making it possible for the plant to adapt to the environmental stress. For example, when water is limited, hormones signal for plant growth to slow down. This is one of the reasons crop yields drop during times of drought. In this activity, we'll learn about plant hormones and how they affect plant development and growth. We'll learn how plant functions are controlled by their biological clocks and the interplay between environmental cues and internal processes. We'll also see how plants alter their survival strategies in response to environmental stresses such as drought and plant pathogens. What are plant hormones? Plant hormones are a lot like animal hormones. They are chemical messengers that control how a plant grows. They are generally organic molecules made in one part of the plant and then transferred to another part of the plant where they act. For example, when a seedling emerges from the soil, it grows toward the light source. How does it know where the light is? The coleoptile is a protective sheath covering the shoot and seed leaf of grasses. Early experiments demonstrated that when this tip is cut off, the stem no longer bends toward the light. If the tip is placed on agar, so that any chemicals present in the tip can leach into the agar, then contact with the agar alone causes the plant to bend. These and other experiments demonstrate that a chemical in the tip of the coleoptile is responsible for the bending of the stem. The chemical is indole-3 acetic acid, an auxin hormone. Auxins are compounds that induce cell elongation and other plant activities and chemically resemble indole-3 acetic acid. The directional growth of a plant in response to light, such as their bending toward it, is called phototropism. In some plants, exposing the coleoptile to light causes the phosphorylation of photoreceptors on the plasma membrane. The phosphorylation initiates a signal that causes auxin to accumulate on the shaded side. The higher concentration of auxin on the shaded side causes the shaded side to grow faster, and the plant bends toward the light. Plant stems grow up and plant roots grow down. They both display gravitropism, the directional growth in response to gravity. If the coleoptile is placed on its side, increased growth occurs on the lower side, causing the tip to curve upward. The increased growth results from an increase in auxin concentration on the lower side. Scientists think specialized cells, called statocytes, are responsible for sensing gravity. Statocytes contain dense starch grains or other solid particles. Experiments conducted on space flights have suggested that the settling of these dense particles in response to gravity enables the plant to sense the downward force of gravity and alter its auxin concentrations and growth accordingly. Auxins are also involved in thigmotropism, the directional growth of a plant in response to touch. Thigmotropism is seen in climbing vines and in the curvature of plants around rocks and other solid objects. Another important class of plant hormones is the gibberellins. Gibberellins have 19 or 20 carbons that form 4 or 5 carbon rings. Gibberellins are named after the fungus Gibberella fujikuroi, which causes rice plants to grow too fast and die. The Japanese scientist Eiichi Kurosawa demonstrated that this fungus causes the rapid growth of rice plants by secreting a chemical now known as a gibberellin. Later, it was demonstrated that plants themselves make gibberellins. When a mutant plant with short stems is sprayed with gibberellin, it grows to its normal size. Gibberellins affect most aspects of plant growth. In some plants, they break seed dormancy and promote germination. They also stimulate flowering and the growth of fruit. These effects are often interconnected. For example, the gibberellin produced in grape seeds stimulates the growth of the fruit. For this reason, seedless varieties of grapes are smaller than grapes with seeds. Today, nearly all of the seedless grapes we eat are sprayed with gibberellin to promote their growth. The hormone ethylene causes fruit to ripen. Ethylene is unique in that it is the only hormone found in gaseous form. Bananas ripen faster when placed in a bag because the bag traps the ethylene gas given off by the bananas. In addition to inducing ripening, ethylene promotes the plant's senescence, or aging, that generally occurs at the end of the growing season. One of the reasons that leaves drop off in the fall is because ethylene concentrations in the leaf tissue rise and auxin concentrations drop. Unlike ethylene, which promotes plant senescence, the cytokinin hormones delay plant death. They stimulate cell division and are often added to stimulate shoot and bud formation in plants grown in tissue culture. Cytokinins are the only hormones known to be found in both plants and animals. They are derived from the base adenine, the same base found in adenosine triphosphate, or ATP. The hormone abscisic acid, or ABA, is a plant growth inhibitor. It induces dormancy and prevents seeds from germinating. ABA is a 15-carbon compound produced in chloroplasts and other plastids. Water stress induces the production of ABA, which causes the stomata to close. Many different plant hormones work together to control plant growth and development. Now that we've seen how plant hormones control growth, let's see how growth is controlled by a plant's biological clock. Most forms of life have an internal means of measuring time, and plants are no exception. Plants respond to 24-hour cycles called circadian rhythms. Circadian rhythms control gene expression, leaf and petal movements, the release of floral fragrances, the opening and closing of stomata, and many of the metabolic activities associated with photosynthesis. For example, stomata generally open during the day to allow carbon dioxide in for photosynthesis and close at night to minimize water loss. How do stomata know when to open and when to close? Stomatal openings and closings respond to numerous external cues such as light and carbon dioxide concentration. In addition, an internal clock keeps track of the 24-hour cycle so that stomatal opening and closing occurs on a 24-hour cycle even when the plant is grown in continuous light. While some of the molecular details of stomatal opening and closing are well known, such as the involvement of the hormone abscisic acid, the molecular basis of the internal clock remained a mystery until just recently. Scientists had suspected for over 10 years that oscillations in intracellular calcium concentrations played a role in the plant's internal clock mechanism. Calcium is a common second messenger in many signal transduction systems. In June of 2001, researchers at the University of California, San Diego, and in Munich, Germany, reported that they cracked the code embedded in the calcium signals of a plant. Using a mutant form of the plant Arabidopsis, they discovered that specific frequencies of calcium oscillations signal the opening and closing of stomata for extended periods of time, such as in their closure at night. Circadian rhythms also control the movement of leaves. The French astronomer Jean-Jacques Ortu de Morin was intrigued by the daily opening and closing of the leaves of a plant, so he did an experiment. He started with the hypothesis that the plant opened its leaves during the day in response to light, so he placed the plant in the dark. Based on his hypothesis, he expected the leaves to stay closed while the plant was kept in the dark. To his surprise, he observed that the leaves still opened on a daily basis even though they received no light. This experiment, done in 1729, is the first experiment known to study the circadian rhythms of a plant. Some flowers, like morning glories, bloom only during the day. Others, like evening primrose, bloom exclusively at night. The release of floral scents also depends on the time of day. Many flowers have co-evolved with the animals that pollinate them. Flowers pollinated by nocturnal creatures such as moths and bats tend to be open at night. Flowers that depend on daytime pollinators are open during the day. The response of a plant to the relative lengths of day and night is called photoperiodism. A single exposure to light during an extended dark period may shift any circadian rhythm. For example, florists control photoperiodism and trick greenhouse plants to bloom out of season by exposing them to artificial light. Blue-green pigments found in plants called phytochromes become activated when light is present. The active form of phytochrome influences plant hormones, which then influence plant development, such as flowering. Seeds also contain phytochromes. If a tiny seed germinates very deep in the soil, it won't have enough energy to grow to reach the surface. So seeds don't germinate until the phytochrome they contain is activated by light. Alternatively, if a seed is under a deep forest canopy, it won't germinate until an opening in the canopy forms. The opening allows sunlight to reach the seed's phytochrome and activate it. The responses of plants to their environment are fairly predictable under favorable conditions. But how does environmental stress alter these responses? We'll find out in the next section. To carry out photosynthesis, a plant needs water and carbon dioxide. Carbon dioxide enters a plant through open stomata at the expense of water, which exits whenever the stomata are open. When water is limited, signals are sent to close the stomata. These signals involve hormones. For example, during times of drought, levels of the hormone abscisic acid rise. The increased level of abscisic acid alters calcium oscillations within the plant, and the stomata close. Have you ever wondered why an overwatered houseplant dies? One of the reasons may be that it has suffocated. Just like animals, plants use cellular respiration for energy. The process of breaking down stored foods, such as starch, requires oxygen for the oxidative phosphorylation of ATP. Roots absorb oxygen from air spaces in the soil. When excess water is present, these spaces are filled with water, and the roots are unable to obtain enough oxygen to meet their energy needs. Eventually, the plant may die. High salt concentrations can be toxic to plants. Excess salt lowers the water potential of the soil, which reduces the ability of the plant roots to absorb the water. Recall that in osmosis, water potential predicts the direction water will flow based on solute concentration and applied pressure. Water molecules tend to flow from an area of low salt concentration to an area of high salt concentration. Plants respond to the soil's lowered water potential by making solutes of their own. This keeps the water potential of their cells more negative than that of the soil, ensuring that water can flow in while salts stay out. Excess heat stresses plants by damaging their enzymes. Metabolic processes, such as photosynthesis, are carried out by enzymes. Enzymes typically are proteins that denature or lose their tertiary structure at high temperatures. Plants react to high temperatures by producing heat shock proteins. Heat shock proteins facilitate the refolding of proteins back into their native tertiary structures. Excess cold damages plants by disrupting their cell membranes. A cell membrane consists of a lipid bilayer through which proteins and lipids can move. When temperatures drop, the lipid bilayer becomes less fluid and may break if ice crystals form. Plants adapt to low temperatures by changing the composition of their membranes. They add more unsaturated kinked lipids that do not pack as well as saturated lipids. Unsaturated lipids kink because they have double bonds. The kinks allow the membrane to remain fluid at lower temperatures. Another strategy plants use to protect themselves from the cold is to move water out of their cells. This concentrates sugars and other molecules and lowers the freezing point of the remaining solution inside the cell. In addition to the environmental stresses associated with drought, floods, salinity, and temperature, plants have to defend themselves against plant-eating animals and disease-causing organisms. To protect themselves against plant-eating animals or herbivores, plants have evolved bitter-tasting chemicals, toxic chemicals, and mechanical defenses such as thorns and thistles. Like plant defense against herbivores, plant resistance to pathogens is also based on physical and chemical barriers. Infection of a plant by fungi, viruses, and bacteria may harm or kill it. To prevent infection, plants have developed physical barriers such as their epidermis and deposits of callose and lignin. When a plant's physical barriers are breached, chemicals may be used to fight off the invading pathogens. Some plants respond to a pathogen attack by producing toxic compounds called phytoalexins. Phytoalexins have general fungicidal and bactericidal action. Plants also produce specific chemicals for defense. For example, some strains of wheat carry a gene that offers them protection against the fungus pathogen that causes leaf rust. Why do leaves turn colors in the fall? They respond to a complex mix of signals involving hormones, pigments, length of daylight, temperature, moisture, and genetic traits. Plant hormones are chemicals that affect seed germination, growth, flowering, reproduction, fruit formation, and senescence or plant death. There are 5 major types of plant hormones: auxins, gibberellins, ethylene, cytokinins, and abscisic acid or ABA. Auxins, gibberellins, and cytokinins promote growth, whereas abscisic acid and ethylene inhibit growth and promote senescence. Plants have evolved for optimal survival in their environments. They often have co-evolved with the animals that pollinate them. This has caused them not only to develop structures that facilitate pollination, but also behaviors that improve their chances of being pollinated, such as only opening their flowers when pollinators are active. Plants also have evolved ways of adapting to changing environmental conditions. They can respond to changes in moisture, temperature, salinity, and to the presence of herbivores and pathogens.